We've been discussing reproductive isolation and its role in speciation using the definition of biological speciation, which indicates that two groups are considered new species when they can no longer interbreed and produce viable fertile offspring. We defined several species concepts, and then we talked about pre-zygotic isolating methods. So now we are going to move on to post-zygotic isolation methods. A post-zygotic barrier is a barrier that inhibits a hybrid zygote, so an organism from two different species whose egg has been fertilized by a sperm and an individual has been produced. We're going to talk about how post-zygotic barriers will inhibit that organism from developing into that viable fertile offspring. There are three main types here, and they are basically levels of viability and fertility. So first we have reduced hybrid viability, then reduced hybrid fertility, and hybrid breakdown. And with all of these, this is post-zygotic, so fertilization has happened. So in all of these, there is an egg that develops to some extent into an organism. So the fertilization has happened. The block or the reproductive isolation is happening at any point after that fertilization. So we could also have an extreme case of reduced hybrid viability where the egg is fertilized but never develops farther than that. First, we have reduced hybrid viability. So viability, basically a viable organism, viability means healthy and able to grow and develop properly. So reduced hybrid viability means that the development or the ability to survive in this as this hybrid organism might be reduced. So this happens because genes from the two separately isolated parent species may interact, may cause problems. The organism may not develop correctly. So our example here is a group of California salamanders. We've got a picture of a single salamander here. These are called N-Satina salamanders, and there are a variety of them that grow and live in California. When different species, different groups of these salamanders interbreed, they're not as hardy or as healthy as the individuals who come from interspecies, meaning within the same species, interbreeding. So these guys rarely survive because they're not as hardy, they're not as viable. So as you get these different groups, different types of salamanders, they may have different levels of viability. They may not complete development, or even if they do, they might be frail and unhealthy, and so they are very unlikely to leave offspring of their own. Because remember, part of the biological species concept is that they can then leave offspring. If they're not healthy enough to leave offspring, then these two groups are considered to not be the same species. If their offspring are not healthy enough to make offspring, then they're considered separate. Next, we get to reduced hybrid fertility. This is probably the mechanism you are most familiar with. This is the mechanism through which we get mules, we get ligers, we get a variety of other organisms. Azores is another common one, not super common, but something you might be familiar with. But you're probably all familiar with mules. Mules are the offspring of a donkey and a horse. So these guys are perfectly viable. They're often vigorous, strong animals. They're used a lot for hauling heavy loads, pulling heavy loads, etc. And so they are absolutely functional organisms. The problem comes up when we get to the fertile part. So the mule, or the hybrid in this case, is infertile, meaning they cannot produce their own offspring. The difference is that the chromosome number or structure differs so that pairing doesn't occur correctly during meiosis. So when gametes are formed for those mules, their gametes don't contain a balanced number of chromosomes, and so they can't combine with the chromosomes from a donkey, they can't combine with chromosomes from a horse, and they often have an odd number or an unusual number, and so they can't combine with chromosomes of their same group. So these guys, to illustrate this example, the horse has 64 chromosomes, the donkey has 62, and when fertilization happens, meiosis happens, and the sperm and the egg come together, the donkey ends up with 63 chromosomes. I'm not going to show you a picture of how that works for meiosis. If you want to try to figure that out, that's perfectly fine. If you want to just accept that that odd number doesn't pair up correctly during meiosis, that's perfectly fine. So you can't get a viable sperm or egg. So these guys are almost always sterile. I believe that I forgot to talk about the growler bear. Way back in the beginning of the lecture, there are pictures of growler bears. This is another hybrid that is becoming more common. Growler bears are a cross between a polar bear and a grizzly bear, sometimes called Pisleys. These guys are a little bit different. The growler bears or the Pisleys actually are sterile, and they do reproduce in the environment, in a natural environment. They can breed, the growler bears can breed with a grizzly, they can breed with a polar bear, and they can breed with another growler bear. So they do not fall into this category. They are actually both viable and fertile. Lastly, we have hybrid breakdown. Now, this is kind of a combination of the two. In hybrid breakdown, the first generation of hybrids are perfectly viable and fertile, but then their offspring are not. And so because their line can't continue, because their lineage cannot continue, we say that this is a postzygotic barrier. So the first generation is great. Second generation is feeble or sterile or non-viable and infertile. Usually this is due to something called polyploidy, but it could potentially arise from any situation where gametes are formed and have an incompatible number of chromosomes. So chromosome differences can arise in a couple of ways. You can have a difference in chromosome numbers, like our horse, mule, and donkey example, or you can have something called polyploidy. Polyploidy is a really quick way to induce speciation. Polyploidy is where the entire genome of an organism is replicated. And I don't mean duplicated as in DNA replication, but what I mean is a fertilization event happens and the chromosomes don't separate correctly, and so all of a sudden now you have a cell with double the amount of chromosomes, maybe 4 copies instead of 2 copies of all of your chromosomes. This can happen by autopolyploidy, which is where the same species has a duplication event. So 2 organisms come together, their chromosomes don't properly separate, and now you've doubled your chromosomes. Or allopolyploidy. Allo means other. And so in allopolyploidy, you get the combination of 2 genomes, say one wheat genome and then another wheat genome, but they are incompatible. They don't separate correctly during meiosis, and now you have double but of different genomes. So now all of a sudden you have this chromosome incompatibility, and so even if your first round is still viable, the second one may not be. Figure 20.2 in your textbook has a really great summary of all of these. You are welcome to check that out. I put it here for easy reference, but you can also go back to your ebook and look at that. So here's an example I would like you to think about. This is the western meadowlark and the eastern meadowlark. So their ranges are generally eastern half of North America and Central America and western half of North America and Central America. You can see there is some overlap of their range in the middle. So these guys have two very distinctive songs. They're really pretty songs. I grew up in Southern California, and we could always hear western meadowlarks singing in the fields around our house. I really love them. Eastern meadowlarks are just as pretty, but their call is a little bit different. So I'd like you to think about what kind of isolating mechanism has led to speciation between these two meadowlark groups.